Aliya Larsen grew up here on the Oregon coast, so she's a true local and spent her high school summers volunteering at the Oregon Coast Aquarium. She's now a student at Carleton College in Minnesota, majoring in mathematics with a minor in Spanish. She's especially interested in science and plans to pursue a master's degree in biostatistics after graduation. Outside the classroom, Aliya is also a standout athlete. She competes in varsity cross country and track, and was part of the team that won the national cross country championships in 2023. So she is very speedy. She's got two dogs named Whidbey and Jasper, and she also really loves reading fantasy. We're so excited to hear from her today. So please join me in welcoming Aliya. Over to you, Aliya.
Thank you. So today I'm going to be talking about my project through the Cape Perpetua Collaborative called Exploring the Benefits of a Marine Reserve. A little bit about me. As previously said, I'm a senior math major and Spanish minor at Carleton College in Minnesota, and I grew up here on the Oregon coast. I spent my summers in high school volunteering at the Oregon Coast Aquarium, and this is also my second year working on this project. So we'll get into that a little bit more later. So this is what I'm going to be talking about today. I'm going to first go over the history of the Cape Perpetua Marine Reserve, and then discuss some of the details of my project. And then I'm going to get into the 2025 data analysis on my tide pools, and also then I'm going to compare my analysis this year to last year. First, I'm going to talk about the Cape Perpetua Marine Reserve history. So in 2013, Cape Perpetua, along with the 4 other Oregon locations, became a marine reserve. Cape Perpetua is the largest marine reserve on the Oregon coast, and it has been one for 12 years. This is important because it can take it can often take decades before full effects of a marine reserve are noticeable on the populations in the ocean. And some of these factors that make these changes take longer include species interactions, climate change, and the age of maturity or age of reproduction for various species. I tried to find data on when the Cape Perpetua Marine Reserve was originally established to try to compare it to what I found now, but I was unfortunately unable to do that. So going off of the age of reproduction for species, this is really important because that determines like when populations begin to change and fluctuate with new generations coming into the marine reserve. So I gathered some of the data on when these species, the main species I collected data for, begin to reproduce. So a sea star is 5 years, and anemones can take 5 to 10 years depending on the species. Purple sea urchins, 2 years, tidal sculpins, 1 year, limpets, 9 months to 3 years, also depending on the species, turban snails, 1 to 2 years, dog whale, 3 years, rock crabs, 1 to 2 years, and Dungeness crabs, 2 to 3 years. So now I'm going to get into the details of my actual project. So first, my research question that I was studying this summer was how does the biodiversity and abundance of species differ in the tide pools in the Cape Perpetua Marine Reserve compared to the tide pools in the non-marine reserve of Seal Rock during the July and August of 2025. An important thing to note here is biodiversity refers to the number of different types of species, whereas abundance refers to the number of each species. Now I'm going to talk about some of the words or phrases that I'm using and kind of why I chose this specific research project in questions. So first of all, tide pools are found in the rocky intertidal zones, primarily on the west coast. Many creatures are lashed onto the rocks to survive the fluctuation of the high and low tides. and you can often see them at low tide when the water goes out. And then high tide brings in the fresh nutrients and food sources to the animals that are living in these tide pools. A marine reserve, which is what Cape Perpetua is a protected area that prohibits the removal of marine life and resources, and also prohibits any development in the area. They're often primarily just used for education and research purposes. So Cape Perpetua is a marine reserve, which is why I picked that as my first location to study. They have stronger protections on tide pools areas, which should mean that you see more biodiversity and abundance of species in this area. And then Seal Rock is the other location I picked to conduct my research on. And this is not a marine reserve or protected area. It has public beach access and is a very popular tourist location to go tide pooling. So it sees a lot of foot traffic. And then for my data collection, I collected data twice a week at all of my selected tide pools at Cape Perpetua and Seal Rock. And I used the same tide pools that as the ones from last year. And I recorded every different species I saw in each tide pool and counted how many there were in each tide pool, and also took general observations of the conditions each day. Now we're going to get into my tide pool data analysis for this year. So since I was using the same tide pools as I did last year, I want to take a look at how they compared to the conditions from last year. So first at Cape Perpetua, these were the 3 tide pools I used this year and last year for all of my data collection. They showed pretty much no difference throughout the 2 years, so there's nothing really much to say on that. However, Seal Rock showed a drastic change from this year to last year. The water levels seem to be a lot higher this year, and the areas where tide pools used to be shifted up onto the rocky areas because of this. And because of this, I really didn't see much in the tide pools in the same tide pools that I collected data from last year, because it was pretty much all just within like the ocean currents at this point. So because of this, I decided to collect data from a 4th tide pool at Seal Rock, one that had more of the biological diversity that I was looking for. And I did a one-on-one comparison to tide pool 2 at Cape Perpetua because they had pretty similar sized tide pools. So all the graphs that we'll be analyzing for this section of my presentation is going to be only looking at the species and abundance that I saw in tide pool 2 at Cape Perpetua and tide pool 4 at Seal Rock. So the first species I looked at were green anemones. These were probably the most common species I saw in the tide pools, both at Cape Perpetua and Seal Rock. They use their tentacles that you can see on the slide to stun their prey, and you can actually see the one in the left of the photo that is eating. And then these are the results from my data collection this summer. These are all the different days I went out and collected data on the bottom, and then on the left side of the graph refers to how many I saw each day. So overall, you can see that I saw more green anemones in Cape Perpetua compared to Seal Rock. And at the bottom of the screen, I have a p-value to demonstrate significance. And the difference in the amount of green anemones at Cape Perpetua and Seal Rock from this graph was indeed significant. The next species I looked at were aggregating anemones. They are often closed up at low tide, so they're a little harder to spot. They reproduce by cloning, so they're often found in packed groups, which is interesting. And overall, I saw more at Cape Perpetua. You can see on day 10, there's a massive increase in aggregating anemones in Cape Perpetua in tide pool 2. And I'll talk about this a little bit later, but there's a lot of seafoam in Cape Perpetua that covered the tide pools. And this day, it had kind of all cleared away, so I was able to see a lot more, and which caused this massive spike in the amount of aggregating anemones I saw. And these results were also significant. The next species I looked at were tidepool sculpin. These are tiny fish that can be found in the tide pools. They are sometimes eaten by the green anemones, but are mostly prey to larger fish at high tide and birds. And you can see them in the picture circled. They're a little hard to spot sometimes. And overall, it kind of varied where I saw the tidepool sculpin, and compared to Cape Perpetua and Seal Rock. Some days there were more in Cape Perpetua, and others there were more at Seal Rock. Since they aren't really latched onto the rocks as much, it can definitely vary from day to day where they will be and how often you can see them. Because I think these and the crabs were the ones who were the only species I looked at that weren't latched onto the rocks like most other tide pool creatures are. So these results were not significant. The next species I looked at were limpets. There's I think two circled in the photo here, and they have a muscular foot that allows them to cling on the rocks with very strong force, even though they are pretty small. And overall, I only saw limpets in Cape Perpetua. I didn't see any in Seal Rock in Tidepool 4. So these results were significant. The next species I looked at were turban snails. An interesting fact about turban snails is the shell grows with the snail, which creates rings around the door opening, and other creatures can live in the shells after they die, like hermit crabs. Turban snails, I mostly saw them in Cape Perpetua, only saw one in Seal Rock one of my data collection days, so these results were significant. And then I next looked at crabs. I mostly saw rock crabs, but also hermit crabs, a couple hermit crabs, Dungeness crabs, and kelp crabs. But I combined them all into one category for the sake of the graphs, because I didn't really see as many of the other groups. And you can see the crab in this photo that's circled. I overall saw more crabs in Cape Perpetua, and these results were significant. Dogwelk is the next species I looked at, and they are a lot of times very common on the Oregon coast. They are interesting because they're pretty small, but they often eat mussels and barnacles by drilling in and liquefying their food. And for dogwelk, the first half of the summer, I tend to see more in Seal Rock, and then the second half more in Cape Perpetua. So overall, these results were not significant because of the variation between when I saw them in Cape Perpetua compared to Seal Rock. Now looking at biodiversity for Cape Perpetua, Tidepool 2, and Seal Rock, Tidepool 4. In Cape Perpetua, I saw 14 different species, and in Seal Rock, I only saw 10 different species throughout the summer. So these are the general findings from my results comparing Tidepool 2 at Cape Perpetua to Tidepool 4 at Seal Rock. Overall, the green anemones, aggregating anemones, limpets, turban snails, and crabs were significant at a 5% level. This means that these results were not by chance. We don't necessarily know why there was a difference in how many I saw compared to Cape Perpetua and Seal Rock, just that the abundance of these species is significantly greater in Cape Perpetua compared to Seal Rock for whatever reason it may be. Tidepool sculpins and dogwelk showed no significant difference. And then for biodiversity in Cape Perpetua, I saw Dungeness crabs, kelp crabs, and a sea star, whereas in Seal Rock, I didn't see, and also I believe hermit crabs, whereas in Seal Rock, I didn't see any additional species aside from the ones I showed in my graphs. And then for abundance, the statistical testing demonstrated that the abundance of species was significantly greater in Cape Perpetua compared to Seal Rock. aside from the tidepool sculpins and dog whale, which showed wide variation. Some limitations to this project was the shorter time frame and then the conditions, as I previously stated, in Cape Perpetua, like the sea foam made it difficult to see into the tide pools at times and get a proper estimate of how many species were in the tide pools. And also only being able to compare one tide pool at each location was difficult because of the shifted landscape in Seal Rock. So now I'm going to get into the comparison to my 2024 data collection, which will address a couple of these limitations. So for my comparison to 2024, I did not use tide pool 4 from Seal Rock. I just used tide pools 1, 2, 3 from Cape Perpetua and tide pools 1, 2, and 3 in Seal Rock from in both 2024 and 2025. So the first species I looked at again were the green anemones. Again, this is tide pools 1, 2, and 3 from Cape Perpetua and tide pools 1, 2, 3 from Seal Rock in both 2024 and 2025. And so I overall saw a lot more in Cape Perpetua both years, the most being last year, and then none in Seal, or I think 1 or 2 in Seal Rock in 2025. Aggregated anemones, I saw the most of them in Cape Perpetua of 2024, followed by Seal Rock from last year as well. For typal sculpins, I saw the most in Cape Perpetua this year, actually, and then followed by Cape Perpetua from last year. For limpets, I saw the most in Cape Perpetua last year and less than 5 in Seal Rock both years overall. For turban snails, I saw the most in Cape Perpetua this year and not really very many in Seal Rock both years. Crabs, I saw the most in Cape Perpetua this year and followed by Cape Perpetua from last year. And then dog whale, I saw the most of in Cape Perpetua in 2024, followed by Cape Perpetua this year. And again, not very many, less than 5 in Seal Rock both years. So now getting into the biodiversity of Cape Perpetua and Seal Rock, only looking at tide pools 1, 2, and 3. In Cape Perpetua both years, I saw 15 different species. There was some variance in what the species were, which I'll talk about in the next slide. And then in Seal Rock in 2024, I saw 13 different species, so 2 less than last year, but still pretty comparable. Whereas in 2025, if I exclude tide pool 4 from my data collection, I only saw 3 different species. So you definitely see there was a massive drop-off in the species in Seal Rock because of the changing conditions, which can definitely make it harder for species to thrive in this ecosystem. So overall, for my statistical significance in abundance, in 2024, the green anemones, limpets, and dog whelk were significant at a 5% level. It looked like there was a lot of variance between Cape Perpetua and Seal Rock in some of the graphs. However, when the data was split up week to week, there was a lot more variance. And compared to Seal Rock and Cape Perpetua, which is why only a couple species were significant. And then in 2025, all species were significant when you compare Cape Perpetua to Seal Rock because I pretty much saw I think I only saw 3 different species and very few of those at that. And for biodiversity in Cape Perpetua last year, I saw a sea urchin and shore crabs which I did not see this year. And then this year, in 2025, I saw Dungeness crabs, a sea star, and what is interesting is I saw a lot more species in tide pool 3 at Cape Perpetua compared to last year. Last year in tide pool 3, I believe I only saw some bay mussels and a couple sculpin. However, this year I got to see some anemones coming into the population in tide pool 3, which was exciting to see. And then for abundance in Cape Perpetua, I saw more green anemones, aggregating anemones, limpets, and dogwelk in 2024, whereas in 2025, I saw more tidal sculpin, turban snails, and crabs. Obviously, for the biodiversity and abundance of species in seal rock, I saw a lot more of in um, I saw a lot more in 2024 compared to 2025 because of the shifted landscape. I really wasn't able to see that much this year, and if you exclude tide pool 4. So some limitations to this are the shifted landscape and tide and seal rock doesn't really fully give you an accurate representation of the biodiversity and abundance of species in the whole beach area. So the most abundant locations had changed across the beach. However, we can see that the consistent landscape at Cape Perpetua, how that might make a better environment for tide pools and tide pool species to continue to grow, as we saw in tide pool 3 at Cape Perpetua, and to continue to grow and thrive. So overall, I did find a higher abundance and biodiversity of species at Cape Perpetua both years. This could be from a variety of reasons, however, and this project did have its limitations. However, it is true that the marine reserve and the protected area was the place with the most biodiversity and abundance of species in its habitat. The it also needs to be said that Cape Perpetua was chosen to be a marine reserve because upwelling in the area creates more nutrient-rich waters for species to thrive and produce lots of offspring. So this could inherently be why there's also more biodiversity and abundance of species in Cape Perpetua because of this upwelling and nutrient-rich waters. But this also means that preserving these areas are important, so the protections are still helpful. There is also further research on marine reserves at the Partnership for Interdisciplinary Studies of Coastal Oceans. They have great research projects based on marine reserves around the world, and I based some of my research and finding on, or I based some of my research on some of their previous studies. So that's a great resource to also check out if you're interested in learning more about marine reserves. And then these are some of the sources I used. Questions?
Awesome. Thank you, Aliya. Such a like a meaty project. Like so much data that you collected and cool observations that you made. I always just to our listeners, I had Aliya text me when she was about to go out tide pooling. So I'd get a text from her saying she's heading out to Seal Rock or Cape Perpetua, and I was always curious what she might find that day. So it's really great to see what her results are. Feel free to drop any questions you have in the chat. In the meantime, I will ask a couple of questions that came to mind in our last few minutes that we have. I think, Aliya, your project is so interesting and it really encapsulates some challenges that scientists have when collecting data out in the real world, notably your tide pools from Seal Rock, tide pools 1, 2, and 3, and how the changing conditions affected what observations you were able to make. And those are things outside your control, and it's a very common thing in a lot of research projects. And if you were to do a project like this again, keeping that sort of challenge in mind, what do you think, what would you do if you were to do this project again? Would you have more locations, more years of data? I'm of course, every scientist wants the capacity to collect more information, but um, yeah, what would you think? What would be your suggestions for a next iteration of this study?
I think definitely collecting data more years into the future would be interesting to see, even like a decade down the road. And then it could also be interesting to look at a location not on the marine reserve that's a little closer to Cape Perpetua and maybe more of a rocky area compared to seal rock. So that there's less of a chance that the sand and tides are going to change the landscape.
Yeah, totally. For those not familiar with the coast, Seal Rock is it's about a I don't know, it's about 20-25 minute drive north of Cape Perpetua. So there's a bit of distance and maybe like 40 miles, something like that, maybe less. Yeah, so I agree. I think finding a spot closer next time
Mhm.
it'd be better to be able to draw comparisons between the two areas since there wouldn't be the potential for other variables to get mixed in there. But you also have to work with what you have too and have access to as well. So I think you did a really great job with the resources you had. I have a question here that came in from Linda Yoder. She says, wonderful. Does the presence of sea stars indicate a recovery from the wasting disease?
Yeah, I think overall, we've seen, not just through my project, but through other projects at the Cape Perpetua Collaborative, we've seen an increase in sea stars, especially the ochre sea stars in Cape Perpetua and along the Oregon coast. The sunflower sea stars are a different species, and there haven't been as many sightings of them recently, although I do think there have been a couple recently down at Bob's Creek, which is a different type of location.
Hmm.
Um, and those were the species that were most affected by the wasting disease. So um, there have been a lot more in the past decade resurfacing, which is good. Mhm.
Yeah, yeah, that's been some really positive news in the last few years that there have been increased observations of sea stars, of the ochre stars in the tide pools, and then sunflower stars in sort of the deeper areas as well, which indicates that their populations are slowly recovering. They're not near where they were before, but they're slowly recovering, which is great. I think we have time for one more question. So I just love to ask all of our speakers what you're up to next. What's happening next for you career-wise?
Um, I'm going to finishing my senior year at Carleton, and then I hope to apply for master's programs in biostatistics or environmental statistics uh, in the future after that.
Any particular species or ecosystem you want to study in biostatistics?
Um, I'm not really sure yet. Whatever is available and what is needs to be studied after I graduate, I think.
Nice. Great. Well, I know everyone here at the Cape Perpetua Collaborative is really excited to see what you do next, Aliya. I'm sure just based on this work, like which I'll emphasize to everyone, you did almost completely independently, collecting all this data, coming up with the study design and the question and everything. Yeah, I can safely say you're probably going to do very well in a career in biostats. So we are very excited to see what you do next. Thank you everyone for attending and listening to Aliya's talk. Thank you so much, Aliya, for your work and your presentation and everything you've done for the collaborative for the last 2 years. Should also mention Aliya has been one of our star interns and has been tabling at the visitor center and at different events. She was at the Rockfish Day at Oregon Coast Aquarium a few weeks ago, and she's just been a standout intern for us the last couple of years, and we really appreciate your time that you spent with us. So thank you so much.
Thanks for
But
having me.
I think that's everything for today. So thank you and have a good good rest of your day, everybody.